Okay, we can start. Good afternoon, everybody. So welcome to the people here in presence and also to the people in streaming. I'm glad to open this symposium, which is organized by Accademia Medica di Roma in collaboration with the IBSA Foundation for Scientific Research and the PhD program in Morphogenesis e Ingegneria Tissutale. So, as you know, from the title, the forum will talk about the new frontiers in regenerative medicine, and as you know, from the program we will start our forum from the ceremony for the fellowship, IBSA Foundation fellowship, and we call later on the winner of this fellowship. But on behalf also of the president of Accademia Medica di Roma, I would like to welcome all people, in particular our eminent scientists, our speakers, Thomas Rando, Pura Munoz, Feliciano Protasi, and Maurilio Sampaolesi. So thank you very much for coming, to be here. And some little words about the forum, about the topic of the symposium. The symposium, as I said before, will touch important topic such as regenerative medicine. In particular, one of the most ambitious aspirations of modern medical science is the possibility to regenerate every part of a damaged body. And this desire has prompted clinicians and researchers to experiment innovative technologies in order to replace organ and the tissues that are irremediably compromised. So in this symposium, our speaker, our eminent scientist, will discuss the state of art in stem cell biology. In particular, you will learn how stem cells operate and how important also are the macroenvironment in which stem cells should operate. And in particular, take into consideration one of the tissue that has provided a lot of effort to understand the biology of stem cells and how a tissue regenerates, such as skeletal muscle. So our speaker, our scientist today will talk about tissue regeneration and how to try to maintain the tissue in perfect functional activity, and in particular how to rejuvenate the senescent skeletal muscle, senescent tissue. So I would like also to thank my colleagues with whom we chair this symposium, Professor Domenico Salvatore and Andrea Alimonti. And I would like now to call Silvia Misiti, who is the scientific director of IBSA Foundation, who will introduce the winner of the fellowship. So please, Silvia.
Thanks, Professor Musaro. Good afternoon to everybody. And as Professor Musaro said, I have the responsibility to drive this foundation that was created almost 10 years ago from the Institut Biochimique, it's a pharmaceutical company based in Lugano, in Switzerland. And The mission of this foundation is to disseminate disseminate the scientific culture, to organize fellowship activities as what we have done and we do today, workshop, labs in schools, and much more. For this, I invite you to follow us through our website and our social channels, to which we devote a lot of effort. And first of all, I would like to thank Accademia America of Roma to have accepted hosting us for this short ceremony, but very important, because every year we allow this fellowship for young researchers under 40 years old, and this year we received more than 160 applications from everywhere in the world, and this year we are seeing an increasing level of a scientific level. So this year for our scientific board, that is represented in part here from Professor Andrea Limonti, Professor Domenico Salvatore, and Professor Antonio Musarò, was very hard to decide and to select the winners. So after thank Accademia America of Roma for the hospitality, I think it's important that we start with our ceremony, so I called our winners, and I would like to introduce to you each one of them, because they are very top talent in scientific research. So just a few words about the fellowships. This year they are 6, so one in each scientific area, so in dermatology, endocrinology, fertility and urology together, orthopedics and rheumatology and pain medicine, and every year we choose a special edition, and this time we have decided to dedicate this special edition to regenerative medicine. That's why we open this forum, and I think it's a very great opportunity for these young talents to meet such as speakers of today and to meet them. So I would like to start with the scientific area of dermatology, and I call to the podium, that is something like this, Alpaslan Tagusgan. And so would you like to introduce yourself and tell us something about your project?
Yeah, of course. My name is Alpaslan Tagusgan, and I'm a physician scientist, dermatologist by training from the University of Hospital in Essen in Germany. My focus is on cancer metabolism, and what I would like to do with the project is like to better understand how the metabolism in melanoma patients changes during therapy, and how we can use new ways to overcome resistance and help cancer patients or melanoma patients in their therapy. And I'd like to thank the IPSA Foundation for this prestigious fellowship, and I feel really honored to be one of the awardees today.
Okay, thanks. I'll give you our award. As you can see, it's featured, so it's self-explaining.
Thank you so much.
Okay, congratulations.
Thank you so much.
As I said before, we received 160, now I have written here, 160 projects, exactly 105 from women scientists, so it's a very good result, and only 55 men, and exactly from 23 countries. So this was a very good result this year. So the second winner of the day is Diana Farah for endocrinology. Come here, close to the microphone.
So, good morning. I'm Diana Farad. I'm a postdoc researcher in IRFAC, Interface de Recherche Fondamentale et Clinique in Strasbourg, France. And my work aims to study the role of thyroid hormone nuclear receptor, TRF1, in the plasticity of colon cancer stem cells. So I would like to thank the committee, the IPSA Foundation committee, for nominating me. And special thanks go to Dr. Misiti, and I would like to thank also Sarah for all the help and the transport help and everything to be here in Rome. It's my first time, and I like the city and Gracia. Thank you.
Congratulations. And we will wait for the results.
Yeah.
Thanks.
Thank you.
Thank you. Okay, so let's go on with the third one. That is Catherine Tomasek from maybe better from Lausanne. Hello, welcome, Catherine. So present yourself.
Thank you. So I'm Catherine Tomasek. I work at the IPSA, so the Swiss Federal Technical University in Lausanne, working with a group of Professor McKinney. And I want to study how polybacterial urinary tract infections, or interactions basically between different bacteria, shape urinary tract infections and antibiotic treatment outcomes. Because most of the time, bacterial urinary tract infections are not caused by one bacterium, but usually they are caused by multiple bacteria, and we only treat one bacterium, and therefore I'm interested in how the interactions of different bacteria affect the treatment outcome. And I want to thank the IPSA Foundation for awarding me this prestigious award. I think it's very important, especially for young scientists, like for me, like I'm a junior postdoc researcher. It's important to gain experience and knowledge, and such awards they do help in gaining knowledge and to get further in our career. Thank you.
Congratulations.
Thank you very much.
Thanks. I would like to add, just to explain, that our fellowships have to be considered as a salary for our winners, so they have the opportunity to work 1 year granted by us in the same department that they are, or we can help them to change laboratory for 1 year. And as everybody knows in this hall, when you arrive in a new department, you are well known if you arrive already with a salary or part of them. That's why we don't put limits. So some of them live in countries very expensive, so they can add our fellowship to another one. So we hope to give a good substance to these young researchers that are in that delicate phase, between the final, the end of the PhD or the degree, and the time of a permanent position. Okay, so let's go to the special. Now we start with a special edition. For this year, for the special edition, we decided to give 2 fellowships, so we have 2 winners. The first is Marianna Cosentino. That is, so she plays at home, but then she will explain us that she's going also somewhere.
I'm Marianna Cosentino, postdoc at Sapienza University of Rome. So I would thank the IBSA Foundation for this opportunity. My research is focused on the use of a three-dimensional model of muscle engineering tissue for the myocardial infarction repair. And so we want to use this construct and we want to validate and to evaluate as this construct is able to promote morphofunctional rescue after the ischemic injury. And so, thank you everybody.
Grazie Marianna. There is a present also for you.
Thank you.
Congratulations. Okay. And the second winner for Regenerative Medicine is another Italian woman. It's Barbara Peruzzi. Barbara, welcome here.
I'm Barbara Peruzzi. I work at the Bambino Gesù Children's Hospital here in Rome. And my project aims at investigating the effect mediated by mechanical stimuli on the generation of cell-derived extracellular matrices, with the final aim to produce patient-tailored tissue engineering and for regenerative medicine. So I would like to thank IPSA Foundation for this great opportunity to establish my career in the field of mechanotransaction and mechanobiology. So thank you very much. I apologize for my voice.
Thank you, Barbara. Thanks.
Thank you.
Congratulations. And we close with, we have now here the sixth winner. She's June Zhu. She's at Harvard Medical School in Boston, and it was much complicated today for her to be here. So she's following us by streaming. And so for the area of pain medicine, orthopedics, and rheumatology, we awarded the project titled Amelioration of Osteoarthritis with Exogenous Circular RNA using Hybrid Nano Delivery Platform. So, ciao, Zoom, and congratulations to you. We'll send you the award. So I would like to conclude just saying that all the projects of our winners will be described on the IPSA Foundation website, so you can see everything, and also we'll ask our winners to update us after 1 year about the results of and to keep in touch with us as your previous winners. So we are creating a very nice group with this, and every year we contact them to know if they are still in research, if they change or something, and also this is to have a good idea about the situation of scientific research in the world. So from this moment, I officially open the new call. 2022 is the edition number 10, and with the scientific board. Can you put the slide of the scientific board so we can explain them? Also, in addition to the three professors that are here, we have also Christian Wolfram from ATH of Zurich, and Professor Bruno Imtum from Hospital of the University of Zurich. They are at home and didn't come here. And together with the scientific board, we decided for the special edition, we selected Healthy AT. So the next year, our forum probably will be on this, and we'll give you news. Okay, now it's time. I please Professor Antonio Muserot to moderate the forum, and I leave the podium.
So, okay, we can start with the symposium, and the first speaker, as I said before, is Professor Thomas Rando from United States, who will talk about muscle regeneration and rejuvenation, a tale of two stem cells, and I give the microphone to co-chair Domenico Salvatore to introduce more properly Professor Rando, and I invite you, Tom, to come here on the podium.
So, good afternoon to everybody. My name is Domenico Salvatore. I'm from the University of Naples, and it's for me a great pleasure to be here and participate to this symposium, and I wish to thank, of course, the IBSA for the opportunity to be here. And it's really a great pleasure for me and a privilege to introduce the first speaker of this symposium. The first speaker is Thomas Rando, that I have on my left side. Thomas Rando is actually the director of the Broad Stem Cells Research Centers at the UCLA in Los Angeles, but his formation is started a few years earlier in Boston. He got his MD degrees at the Harvard Medical School, and then in Cambridge, always at the Harvard Medical School, he took his PhD. Then, after his postdoc, he moved to Stanford in California, where he became a professor of neurology, but also the director of the Stanford Center on Longevity. And of course, this longevity represented the main focus of his interest. Dr. Rando is an excellent scientist and an opinion leader in the stem cell biology, and the main focus of his research and this laboratory is to study the biology and the physiology of the stem cells, particularly the muscle stem cells, and how this biology impacts and interacts with the process of aging and with the disease related to aging. So it's really a great pleasure for me to give the podium, Thomas. Welcome to Rome.
Well, thank you so much, Professor Salvatore. It's my really great pleasure to be here. What he didn't mention is that I had the real honor and privilege of spending my sabbatical here in Rome at this university. And it was many years ago now, but it was a very, very special time in my life and a really wonderful opportunity. I want to thank the committee for inviting me back and IPSA for sponsoring this, and it's a real great pleasure to be here speaking with you today. So I did title my talk initially A Tale of Two Stem Cells, and I was going to talk about both muscle stem cells and also another stem cell in muscle called fibroadipogenic progenitors, but in the interest of time, I'm just going to talk about muscle stem cells today. This is a muscle meeting, so I'm going to talk about muscle, and our lab studies the biology of muscle and muscle stem cells in particular. And one of our great interests along the way has been in the remarkable ability of muscle to regenerate. And muscle is primarily a very stable tissue, but has really the great ability, let's see if I have a mouse here.
Yes.
To regenerate in response to injury. So on the top, you see a cross-section, a normal histologic image of skeletal muscle, and then below that is the image of muscle 1 or 2 days after a total necrotic injury to the tissue. Over the course of the next week or so, one begins to see the formation of these new muscle fibers, or so-called myotubes, and over the course of a few weeks, those cells will grow in hypertrophy and basically restore the normal structure of skeletal muscle. So really remarkable regenerative potential. And at the core of that regenerative potential are these rare so-called muscle stem cells or satellite cells, which is shown, one of these satellite cells is shown here in this whole field of muscle fibers, and it's stained with an antibody against this transcription factor, PAX7, which is a really tremendous marker because it stains all muscle stem cells and only muscle stem cells. So it's really quite a useful marker, and of course we use this PAX7 locus to drive expression of genes and knockout of genes specifically in this population. Now this population, in the absence of injury, sits basically in a dormant or quiescent state for months or potentially even years in mammals. And so I won't talk about quiescence today, even though we studied this in my lab, but I'll mention it a few times as we go. So one of the interests in my lab, as Professor Salvatore mentioned, has been, why is it that as we get older, we repair our tissues less well than when we're young? And that's true of a skin wound or a bone fracture or a muscle injury. And typically in response to an injury, as we get older, we see less effective regeneration and more scar formation. And this is just illustrated in this slide, which shows young muscle on top and old muscle on the bottom. These are from experimental mice. So you have the uninjured muscle looking perfectly normal. After a single injury, or even after a second injury, after several weeks, you see really normal regeneration in the young animal. But in the old animal, you see something quite different. Even after a single injury, one starts to see these little areas shown in white here, of which is really fibrosis and a little bit of scarring. But then after a second injury, it becomes even more dramatic. So as we injure tissues, the stem cells that are there seem less and less able to undergo effective repair and effect good tissue regeneration. So our lab has been interested in trying to understand why this is, but also to ask the question, is there a way that we're able to basically restore youthful function to old tissue? Can we get old tissue to regenerate as well as young tissue? And initially we were quite inspired by the work of Bruce Carlson and John Faulkner now from many years ago, who were doing studies of muscle regeneration and using so-called heterochronic studies, that is using young and old donors and young and old hosts. And what they were doing is basically taking either young muscle or old muscle and transplanting that into either a young host or an old host. The dramatic conclusion from their studies was this, is that it didn't really matter how old the cells from the donor were. They could be young or old. If they were transplanted into a young host, the tissue regenerated very well. Whereas if they were transplanted into an old host, shown here, the regeneration was very poor. So the general conclusion is that it really isn't so much the intrinsic aging of the stem cells, but rather it's the environment in which those stem cells are asked to do the repair that matters. So this environmental influence is largely, or maybe even primarily, a determinant on how effective tissues repair with age. So we were quite inspired by this, so we pursued another heterochronic study, and that is so-called heterochronic parabiosis. So this is again a situation in which the environment of the animal really determines the effectiveness of the tissue repair. So we would either generate these so-called isochronic pairs, so young animals connected to young animals, or old animals connected to old animals, or the interesting partners are the heterochronic pairs, so a young animal connected to an old animal. And in this case, the young animal is exposed to the circulating environment of the old animal and vice versa. The old animal is exposed to the circulating environment of the young animal. And we asked basically, how effective is tissue repair in these parabiotic studies? Well, in the isochronic or control pairs, the young animals regenerate very well. The old animals, you see a large scar formation. But in these heterochronic pairs, in the old animals, when the muscle is injured, they regenerate really just as well, just as effectively as the young animals. Again, saying that the exposure to this young circulating environment is really promoting old stem cells to behave like young stem cells. And this was work that was really pioneered by two different postdocs in the lab at the time, Irina Convoy and Andrew Brack, who both went on to start their own labs, actually both in the University of California system at this point. So Irina was particularly interested in the intrinsic changes that occurred in the stem cells with these heterochronic pairings. So she was looking at the Notch ligand delta, looking at Notch signaling, which is high in the young, goes down with age, but in these heterochronic pairs, it's sort of normalized. So the young become older and the old become younger by these criteria. So Andrew was looking at a functional readout of the stem cells, which is the fact that with age, the muscle stem cells begin to lose their myogenic capacity and start to adopt, at least some of them, a fibrogenic characteristic. So they actually produce more fibrosis than muscle. And so using this assay, again, in the young animals, that's very low. In the old animals, it's quite high. But in these heterochronic pairs shown here and here, again, it normalizes. So the young cells adopt a more aged phenotype, and the old cells adopt a more youthful phenotype. So we initially did these studies looking at muscle regeneration and liver regeneration. A few years later, with our collaborator Tony Weiss-Corey, we looked at neurostem cells and neural function. And then over the next decade, there was a whole series of papers using this heterochronic parabiotic technique, showing that old tissues adopt a more youthful phenotype when exposed to this young circulating environment. So that was very interesting to us and really opened up a whole wealth of studies, both trying to understand what are the factors that are in young blood or old blood that might be mediating these effects, how the cells respond to that environment and adopt kind of an epigenetic change. And so we and others have gone on to study many of these, and I think you'll hear more about this from Professor Munoz in the next talk as well. But of course, parabiosis is an interesting laboratory technique, but it's not very applicable to humans. So we began to ask questions, are there other ways we can restore youthful properties to old cells? And are there, in fact, physiologic interventions in addition to things like genetic interventions and drug interventions that could potentially be used to restore youthful function to old tissues? And so there's been obviously a lot of interest for a long time on the benefits of exercise and physical activity to maintaining youthful properties and in fact restoring youthful properties. And clearly there are a few things that are known physiologically that can delay the onset of age-related diseases compared to physical activity. So we asked the question, is it possible that if we exercised animals, and in particular old animals, could we restore youthful function to those old animals? So I'm going to summarize. This is a paper that was published about 1.5 years ago, so I'm just going to summarize the results briefly. But this is work that was spearheaded by an MD PhD student in the lab, Jamie Brett, and a postdoc in the lab, Marina Arjona. And the experiment was quite simple. We just exercised the mice for 3 weeks. The mice would run on a wheel voluntarily, and after that time we would evaluate the ability of their tissues to regenerate and their stem cell function in the different groups. So this was the first result and really quite striking to us. So what we're measuring here is on the y-axis, it's just a quantitative measure of tissue repair measured histologically. And we were comparing young animals to old animals, either without or with exercise. So as I mentioned here, the young animals show a robust regeneration which declines with age. What was interesting is that exercise had really no benefit in the young animal, but had a profound benefit in the old animal. And the muscle was able to regenerate almost as well as the young animal in response to this single 3-week bout of exercise. So we looked at the stem cells, and what we looked at are genes that go down with age and are upregulated with exercise using just RNA sequencing. And what was striking was the gene that was most dramatically upregulated was cyclin D1. Now this was interesting because cyclin D is typically associated with cells that are proliferating, but this is being expressed now in a quiescent, non-cycling cell. So we were asking, what is it potentially that would be the mechanism by which cyclin D1 would act in a non-dividing cell? And in fact, there's a lot of evidence of cyclin D1 having non-canonical roles, that is non-cell cycle roles, and one of those most well-established mechanisms is a transcriptional repressor. So it can repress the expression of many genes. So what we did is we looked at all the genes that were known to be regulated by cyclin D, that changed with age, and that responded to exercise. And we looked at all those genes, we asked what are the transcription factor binding motifs that are most highly represented in those genes, and really came down to 2 transcription factors. So SMAD3, which is a mediator of the TGF-beta signaling pathway, and REL or REL-A, a mediator of the NF-kappa-B signaling pathway. And I'll talk about the work we've done looking at TGF-beta SMAD3 signaling. So we looked at a variety of data sets and gene expression patterns, and we asked which of these pathways are most highly anti-correlated with cyclin D1 expression. So if cyclin D1 is a repressor, we would express those pathways to be downregulated. And the pathway that was most significantly anti-correlated was TGF-beta signaling, and the transcription factor that was most significantly anti-correlated was SMAD3. And in fact, there had been evidence of cyclin D1 being a repressor of the TGF-beta SMAD3 pathway. So we basically used an inhibitor of this TGF-beta pathway, this drug shown here, administered to old mice, and then looked at a functional readout of muscle stem cells, which is graphed on the y-axis. So the dotted line shows the level of the activity from young animals. It's markedly diminished with age, but if we use this inhibitor of TGF-beta signaling, those stem cells now adopt a nearly youthful functionality. And to ask if this might be indeed related to cyclin D1 signaling, we generated mice that were heterozygous for cyclin D1 in the muscle stem cells alone. And so that sort of phenocopied the effect of aging, a decrease in cyclin D1, and they showed these kind of diminished activity. If we look at the wild type level here, the heterozygous level is low, but if we use the same inhibitor of TGF-beta signaling, so now downstream of cyclin D1, we could restore youthful function to these muscle stem cells. And so finally what we did is we asked, well, what if we took an old animal and we upregulated cyclin D1 specifically in the muscle stem cells, if the problem is that the cyclin D1 level is too low? So we generated a mouse strain that allows us to specifically induce genes in the muscle stem cell compartment using this RTTA-TRE system or tetracycline regulated system. And just shown on the right here is an example of control where we're using GFP expression as a marker. So here's a satellite cell that expresses high levels of GFP in response to doxycycline. And this is a lower power image where we can basically see all of the satellite cells, and only the satellite cells are expressing high levels of GFP. So we used the same system, and we generated a mouse in which we could induce cyclin D1 specifically in the muscle stem cells. So we generated these mice, we aged them for 2 years, and then we gave doxycycline just for a couple of weeks. And again, we looked at this functionality of muscle stem cell function. So again, the young level is shown here, markedly diminished with age, and even after 2 years, just a couple of weeks of induction of cyclin D1, it doesn't restore the old cells back to a completely youthful phenotype, but it dramatically increases their function, even with just 2 weeks of activity. Okay, so this was our model from this work on exercise. We have these young animals here that have high levels of cyclin D1, which suppresses TGF-beta signaling. With age, cyclin D1 goes down in the stem cells, de-repressing TGF-beta signaling, and therefore suppressing muscle stem cell function. With exercise, cyclin D1 is induced and basically restoring kind of a youthful genotype and a youthful phenotype to these old cells. So the one other piece of data from this paper that I want to share with you is we were able to take the serum from an exercised old animal and transfer it to a non-exercised old animal. And when we did that, here we're graphing this, we see this functionality of the muscle stem cells in the old animals, again, increases back towards a youthful level. So this says that exercise is actually having an effect potentially of secreting factors into the blood that are then having an effect on the muscle stem cells. So last year we followed this up with another collaboration with Tony Weiss-Corey, in which we looked at the effect of exercise on brain stem cells, neural stem cells, and brain function. And what we found is that, shown here in these graphs, is that this so-called runner plasma, so these were again running mice, that plasma was effective in enhancing neurostem cell function and also cognitive function. And using a proteomics approach from blood, we found that this protein clusterin is upregulated in the blood in response to exercise and actually can have these effects directly on the brain and the neurostem cells. And we haven't published this yet, but we've gone on to show that this same protein is produced in muscle fibers in response to exercise and is secreted. So it may be this is one of the proteins that's secreted from muscle that may have systemic effects. Okay, so in just the last part of the talk, I wanted to turn to one other physiologic intervention that is known to maintain youthfulness and actually extend lifespan. So in addition to exercise, the field that's probably most well studied in the biology of aging is diet. And there's probably few things studied as well as dietary restriction or caloric restriction in terms of extending lifespan and extending health span. And that's been shown across many species that are basically maintained on a low-calorie diet for their lives. So our question was, like exercise, can we have a dietary intervention, for example, dietary restriction, that might restore youthful properties to old stem cells? What we decided to use, instead of lifelong caloric restriction, we decided to use fasting because it's more controllable and it's probably more amenable to applications to humans than lifelong caloric restriction. So our question was, does fasting itself enhance basically the function or resilience of muscle stem cells? And this is a project being led by a postdoc in the lab Dan Benjamin, and a recent graduate student, Peter Boat. And they asked this simple question, is if we fast mice and then look at the function of old stem cells, how does the fasting affect basically the resilience of those stem cells, or their resistance to dying under various experimental conditions? And so what they found is this one period of fasting in this cell death assay profoundly reduces the cell death or increases the resilience of muscle stem cells. And so just to remind you, when we fast or when we eat a ketogenic diet, what happens is we secrete into the blood these ketone bodies, most prominently beta-hydroxybutyrate, or BHB, which then travels through the blood, gets taken up into cells, and is actually used as an energy source during fasting. So it's converted to acetyl-CoA and enters the Krebs cycle to produce ATP. So we asked, what happens if we just take a ketone body, this BHB, and treat muscle stem cells? And we're looking at an assay in culture here with increasing doses of BHB, the cells have increasing resilience or increasing ability to survive in response to different stresses. If we transplant either BHB or control cells, BHB treated or control cells into a muscle, and these cells are labeled with a fluorescent protein RFP, the BHB treated cells are much more resilient and able to transplant much better than the control cells, and that's just shown quantitatively here. So we had previously shown that with age, there's a marked decrease in the resilience of muscle stem cells, and that in fact, they're susceptible to this form of death called mitotic catastrophe, which is dying during cell division. And this is just shown here from a FACS plot in which propidium iodide uptake is marking dying cells or dead cells, and much higher in the old compared to the young. So we asked what the benefit of this kind of ketosis or BHB treatment might be on these old stem cells. And so we took either young or old stem cells, or actually these are young or old mice, treated with control or ketone bodies, and we asked, how does that affect the propensity of these cells to undergo a kind of mitotic catastrophe when they activate? And here's the young level. Again, the old cells are much more susceptible to this form of cell death, but if the animals are first treated with this ketone body, it reduces this susceptibility from the old level back to the young level. So really restoring kind of the resilience of young cells compared to old cells. So how might this work? How might ketone bodies be enhancing muscle stem cell resilience? Well, in a separate project, we were doing shotgun proteomics, looking for proteins that are expressed specifically in quiescent stem cells. And to our surprise, there was a whole series of metabolic enzymes that are actually quite high in these quiescent cells and go down with activation, as the cells enter the cell cycle. And one of these that caught our eye is this protein OXCT1, because this is the rate-limiting enzyme in the conversion of BHB to acetyl-CoA. So we reasoned if we knocked out OXCT1 in these muscle stem cells, that would prevent the ketone body from inducing this resilience to aged stem cells. But in fact, when we did that, it had no effect at all. So we knocked out OXCT1 in the stem cells, and we showed that this ability of ketone bodies to reduce muscle stem cell susceptibility was true in the wild type cells, but it was also true in the OXCT1 knockout cells. So it seemed as if this normal metabolic activity is not the mechanism by which the ketone bodies are affecting this resilience. So how might that be? If the ketone body is acting via a non-metabolic pathway, what might that be? Well, the most well-established activity of ketone bodies that are non-metabolic is as an inhibitor, endogenous inhibitor of histone deacetylases. And this was published many years ago now by Eric Verdin's group. So we looked at histone deacetylase activity in our cells, and indeed in animals treated with BHB, if we looked at targets of histone deacetylases, they showed upregulation of acetylation. So we're just looking at one target here, lysine 9 of histone 3, that goes way up, but that was true for other targets of HDACs as well. And in fact, if we just used an HDAC inhibitor, this generic inhibitor gavinostat, it phenocopies the effect of ketone body treatments. That is, it reduces susceptibility or enhances resilience of stem cells either in vivo or in vitro. So we're curious to what the targets of HDACs might be that might be mediating this effect. So we did a pan-lysine acetyl Western blot and saw a series of proteins whose acetylation increases by treatment with BHB, and one of those proteins had a molecular weight of 53 kilodaltons. So we hypothesized, surmised that maybe this is actually p53, a gene that we'd studied in detail in response to cell susceptibility with age. And in fact, if we look at acetyl p53, it goes up with fasting or BHB treatment. And in fact, if we look at the gene expression pattern in these cells, there's a very strong p53 transcriptional signature. And I should have mentioned that acetylation of p53 is associated with increased transcriptional activity. And so we tested whether if we knock out p53 in the muscle stem cells, what effect that has, and in fact, it completely abrogates the ability of BHB to induce this resilience. So this is shown in the control cells, and we see a little bit of increase, but no significant increase in cells that are lacking p53. And so finally, we looked at one other strain of mouse, and this is a so-called p53 acetylation mutant that has a series of lysines in the C-terminal region of the protein mutated to glutamine, and so this p53 has a kind of constitutive active form, and in fact, even without BHB treatment, without ketone body treatment, we see this increase in resilience simply by hyperactivation of p53. So this is our model. I'll just end with this. In response to fasting, or I didn't talk about this, a ketogenic diet also works, or direct ketone body treatment, there's an increase in ketone body levels in the cells, and they are metabolized and used for energy, but the cells have also basically adopted this metabolite for another function, and that is by inhibiting HDACs, or histone deacetylases, there's a hyperacetylation of p53, an activation of the p53 pathway, and an enhanced resilience of those cells. So we're now looking at downstream targets of p53 that might be mediating, again, this enhanced resilience. So is this really rejuvenation, what I've talked about today? So we show a restoration of youthful function, youthful molecular features, and it looks as if these cells that are old are becoming younger. But what we don't really have still, I think, is a well-established, well-agreed molecular definition of cellular age. And so these cells, for all extents and purposes, look as if they're becoming younger. But until we have that definition, I think they're still they look younger and they act younger, but we don't really can define them on a molecular basis that way. So let me just end by saying this is my group. This is pre-pandemic, and so many of these people are still up at the laboratory in Stanford, but several have come with me to Los Angeles, and the lab in Los Angeles is growing, and so we're always looking for new students and postdocs. So if any of the students in the audience are interested, let me know. We definitely are recruiting for new postdoctoral fellows. And I want to acknowledge again the people in the lab, both current and former, who contributed to this work. Our collaborator Tony Weiss-Corey, with whom we've done a lot of these studies of stem cell aging and rejuvenation. And of course, I want to acknowledge the support we've received over the years from the NIH, the Glenn Foundation for Medical Research, and the California Institute for Regenerative Medicine for conducting these studies. So with that, I'll stop and be happy to take any questions. Thank you.
So thank you very much, excellent talk. I don't know, there are questions from the audience or from the web? Maybe. Okay, Pura and Antonio. Pura first.
Pura, can you go there so that the people listen?
Any idea of how exercise induces cyclin D1 expression, or is it known how exercise might lead to the upregulation? Is transcriptional or?
So it's a good question. I'll just repeat it. Okay. So the question is, how does exercise induce cyclin D1 in these cells? So we don't know yet. I mean, so we're looking both at proteins in the blood that might mimic the effect of exercise, but also the transcriptional regulation of cyclin D1. We have several candidate pathways, but at this point, we don't know which of these is actually responding to exercise, but it's an important question.
Antonio?
C'è una domanda dalla chat.
So, can you hear me?
Yes, go ahead.
Very nice presentation. And I was wondering if in skeletal muscle the fasting treatment or HDAC inhibitor treatment affect not only satellite cells but other stem cells like FAPs or other population that reside within the muscle tissue?
So thank you for the question. So we have, so what I didn't talk about is that associated with this increase in resilience that I mentioned for the muscle stem cells, there's actually the cells enter what we call a deep quiescence state. And I just didn't have time to talk about that today, but what we've looked at in other stem cell compartments is the response to these kind of interventions to the depth of quiescence. And we do see this in other stem cell compartments, but we have not yet looked as to whether that confers the same kind of increased resilience that we see in the muscle stem cell compartment. We suspect that that will be the case because there does seem to be this correlation between depth of quiescence and resilience, but we haven't looked at it yet.
Okay, thank you.
Yeah, Tom, very nice and intriguing work. So I have two general questions. The first one is very naive, in particular, actually is a curiosity. Aging is a physiological decay. Do you think that, this intervention can be done also when the organism and the muscle is in a way that is already damaged, is already altered? So there is a real rescue of the senescent phenotype, or is a sort of a prevention of the aging? And the second question is related to the second part of your talk. So you demonstrate that the HDAC is one of the pathways involved in this process, meaning that probably that there is a sort of epigenetic clock that can be involved. Can you tell more about this epigenetic involvement?
So thank you, Antonio. So the first question about whether this is more of a prevention versus a reversal, I would say that in fact, if anything, our studies have been more about reversal because we start with mice that are already old, and we exercise those mice or we fast those mice. So the extent that we're seeing a youthful phenotype of the old cells is certainly a reversion. So we're not doing long-term dietary restriction or long-term exercise that would be more of a preventive. So that's encouraging in many ways because it says that you don't have to start these things when people or animals are very young, and that potentially interventions, even late in life, have the potential to restore youthful properties. But it's it's an important distinction. So thank you. In terms of the question of epigenetics, so even though in this study that I talked about, we feel we've identified a target of HDAC that is a transcription factor, so p53, we are very interested in the kind of epigenetic rejuvenation of cells. And so we've done a lot of work to characterize, mostly in the setting of parabiosis, what is the epigenetic signature of old stem cells that are subjected to that kind of intervention, parabiosis. And we see very clear epigenetic changes that occur. We haven't done the same level of analysis in response to either exercise or diet at a global level, but we have done recently, and again, this is just on bioarchives now, we've looked at transcriptional patterns that are across both muscle stem cells, hematopoietic stem cells, and neurostem cells in response to both aging and exercise. And again, there are a lot of commonalities that we think are mediated by common epigenetic rejuvenation. But again, that's a big field that we're very interested in, that I think is where we're seeing this kind of reprogramming of cells to become more youthful at an epigenetic level.
I also have a couple of questions for you. The first one is, you described in the past this cell cycle state of quiescent cells, which is the G alert. I was wondering, what about the cyclin D1 level in those G alert cells? This is the first question. And the second one is a little bit more philosophical, is about your models of circulating plasma into bodies. And I was always puzzled by the fact that during regeneration, but cardiac regeneration, in embryonic life or neonatal life, the heart can regenerate without making a scar. I was wondering whether this kind of model can help us to tell us why in one sense or in the other, to see whether, can you say your opinion?
So again, thank you for those questions. So the first question about cyclin D1 levels in the G alert state, we know that cyclin D1 levels are lower in the G alert state, and that sort of fits with the idea that the more resilient the cell, the higher the level of cyclin D1. Again, I didn't have time to talk about that today, but even though the alert state that we showed is better able to regenerate muscle, it's actually less resilient. So if cells are in this G alert state, over time they decline. So we think that these states of either deep quiescence, which is high cyclin D1, or G alert with low cyclin D1, is related to kind of the ability of cyclin D1 to maintain cellular quiescence, or I should say promote cellular resilience in these states. I mean, your question about the regenerative capacity of embryonic tissues and fetal tissues is really interesting. So I guess the question is really this, is whether the ability of tissues like heart to regenerate during embryogenesis, but then not later in life, is that due to intrinsic properties of the cardiomyocytes and the precursors, or is it due to this environmental influence? I mean, I don't know. I mean, I could speculate that based on our work on the effect of systemic regulators, that there would be a profound effect of the fetal environment in promoting tissue repair, which it clearly does in many tissues. But I don't think that the heterochronic studies, transplanting, that kind of that kind of environment into an older animal has been shown to promote regeneration. In fact, if anything, the heart is pretty difficult to regenerate no matter what in the adult. So I think there might be a combination of the two, but again, I think that's a really intriguing question as to why we lose the ability to regenerate, even tissues like heart or digit tips, things that we can do during embryonic development, but we lose that after birth. But I think that's a critical question.
Thank you. Other questions?
I have a question out of curiosity. Very nice talk. In the first part of your talk, you discussed about the role of the tissue microenvironment in dictating the phenotype that you are observing. However, in the second part of the talk, this was not mentioned, right? Or unless I didn't grasp it. So I was wondering, since cyclin D1 overexpression, the repressed jet data, whether this could affect the secretory phenotype of this muscle cell, and this could end up to have non-cell autonomous effect. So did you try to do your experiment in immunodeficient mice? So exercising, yeah.
So we have not done these experiments in immune deficient mice, and you're right. We never looked at whether induction of cyclin D1 could have cell non-autonomous effects. I mean, the model was based, the bias that we were looking at was based on the fact that the extrinsic factors are inducing this gene, then having the cell intrinsic effects. But absolutely, there's no reason why it couldn't be that there also are secondary cell extrinsic effects. I mean, I would say that, the second part of the talk wasn't only about the cell intrinsic effects, because I mentioned we could transfer the serum from exercise animals to non-exercise animals and induce the effect. So that again suggests that cell non-autonomous, we don't know where the secretome is coming from, but there are circulating factors that are also having effects on the cells.
So there is a question from the audience. Can you come close to here?
Sì, sì.
I could achieve this.
Thank you for the talk. And do you have evidence that cyclin D1 titrate P16 and therefore reduce the senescent-like phenotype of cells?
So we've looked at P16 levels in these cells, and again, I think Dr. Munoz will talk about this. At this age, we do not see these cells expressing high levels of P16 and inducing. So I think we have to be careful about using the word senescence as meaning just getting older, which of course it means, versus the, which I think you're referring to, which is the phenotype of cell cycle withdrawal which senescent has come to mean. So in these cells, they are not senescent because they divide. But we're interested in this connection. But at least at these ages, 2 years of age for mice, we're not seeing these cells entering a true senescent state or having high levels of P16. So we think cyclin D1 is not acting via that pathway.
Thank you. Thank you for your presentation. Do you suppose that extracellular vesicles could be involved in your phenotype because they are producing great amounts of following exercise and they are in the circulation? So maybe you can add.
So that's an excellent question. So when we first did these studies, extracellular vesicles had not been invented yet, or at least they were not called that. So we assumed that the systemic factors we were seeing in the parabiotic experiments were due to circulating proteins, likely. I mean, we didn't really know, but we assumed it was proteins. And we did some early studies like heat inactivation, very simple things that suggested to us that at least some of the effect, the rejuvenating effect of young plasma, was from serum proteins. Having said that, I think we could go back and reevaluate this question exactly in line with your question, is whether there's a component of these effects that are either promoting youthfulness or promoting aging that are mediated by EVs, and I think that's a very interesting area. Thank you.
Thank you for your talk. First of all, sorry for my bad English. I have a curiosity. The aging is a natural phenomenon, so is good to force the organism in the rejuvenation. In other words, have you seen some collateral effects of your study?
So it's an important question. Repeat it.
Can you repeat the
Certainly.
question, because online I cannot listen.
Cannot hear. So the question was that aging is a natural phenomenon, and if I understood it, the question is there any negative consequence of promoting, quote, youthfulness in an aged animal? That's basically the question. And I think it's a very important question. We've thought a lot about this. So most of our work in what I would call promoting youthfulness, whether it's parabiosis or exercise or diet, have been done in a very short time window. And we've not really looked for negative consequences. However, the question remains, and I think this is what your question gets at, if we were to have any of these interventions go on for weeks or months or years, would we have the negative side effect, for example? I mean, can we worry about promoting cancer? If we're promoting sort of proliferative activity of the stem cell compartment chronically as animals age or humans age, is there going to be a negative consequence? And I think that's our main concern. So we haven't done these studies in chronic models enough to answer the question, but it's an important one and always in the back of our mind.
So thank you. It's time now to move ahead. Thank you, Thomas, for your excellent talk and excellent discussion. And I ask Antonio Musaro to introduce the second speaker today. Antonio.
Thank you. So I am pleased also to introduce the next speaker, who is Professor Pura Munoz. So Pura studied pharmacology at the University of Valencia. She obtained a PhD in the Autonomous University of Madrid for work that she carried out at the Script Research Institute at Loyola, the USA. And then she did two different postdoctoral work, one at the University of California in San Diego and another one at the Script Research Institute. Then she moved into the Center for Genomic Regulation in Barcelona, where became a senior scientist, and then in 2008 she moved to the Pompeu Fabra University, basically where she worked until now. And Pura is also a member of the EMBO and the Accademia Europea. She also one of the leaders, like Thomas Rando, in the field of muscle biology. The major research interest of Pura is to understand the mechanism regulating tissue regeneration in aging and disease. So basically we will follow the topic introduced by Thomas Rando. And thank you, Pura, for also for coming here. And the title of the presentation is Muscle Stem Cell Aging, Identifying Way to Induce Tissue Rejuvenation. So I hope that at the end of this symposium we can rejuvenate a little bit, at least at the mental point of view. Thank you, Pura.
Okay. Well, thank you very much, Antonio, and for the kind introduction, and everyone, the organizer of this symposium, ISBA, and the Academia Medica di Roma for having us here. We enjoy twice or three times more than before getting together and seeing old friends and colleagues. So thank you for giving us this opportunity. So yeah, it's very nice and also a challenge to talk after Tom Rando, because I mean, Tom started the field of muscle stem cell aging, and we just followed his pioneer work on parabiosis, and of course this is a huge field, and but yeah, it's good to acknowledge who started the field of muscle aging. This is just a slide to reinforce the concept that with aging, we just have more chances to get diseases. And so one theme we could all think of is if we prolong healthy aging, we might delay diseases. But aging is so, it's such a complex process, it's so polyedric, that it is better to study it with specific questions, with specific models, as Tom said, using skeletal muscle as a model, as a tissue, or as a model, or muscle stem cells. So we also have used through these years, since we started in the aging field, we used mouse as a model system mainly, skeletal muscle as our favorite tissue to study aging, and regeneration. And, but I think that same as Tom, many of the conclusions that we're getting, they don't seem so specific for maybe, we need to prove that, but maybe they're more general for stem cell aging, or at least for quiescent stem cells, and not, so they're particular, we're seeing generalities of decline of mechanisms that are going awry with aging in stem cells. So I will not stay here too much in this slide, just to tell you that this muscle stem cell, called satellite cell, is, as Tom said, in a quiescent state. That doesn't mean dormant or inactive, it's just resting and waiting to be needed to activate and form new fibers if there is an injury or a stress. So this process also is said, so most the destiny of most of the stem cells is to activate, divide, and form in the end new myofibers to undergo myogenesis. Only a subset will go to self-renewal to replenish the quiescent stem cell pool. For most of the talk, I'll talk about satellite cells as a homogeneous population, but it is not completely true. But when we looked at populations of cells, we didn't distinguish them. So work of many groups, I mean, the initial heteroparabiosis experiments that Tom just mentioned, and with Irina Conboy, Andrew Brack, and then other labs in the skeletal muscle field, or the skeletal muscle stem cell field, have shown that both extrinsic and intrinsic changes driven by aging account for the loss of regenerative capacity of skeletal muscle during aging. And we wanted to study, since already 12 or 13 years when we started this, to understand the regenerative decline of skeletal muscle with aging. And in particular, we wanted to study not just aging, but those very light stages of life, that we called geriatric. We thought of calling it senior mice, whatever, but in the end we said geriatric. It was like a way to distinguish mice that in our lab, that doesn't mean that all mice that are 28 months of age that in our lab we call geriatric, that's a geriatric in every mouse, in every laboratory. But for the sake of this talk, or what we've been using in the lab, our nomenclature is that young mice are usually 2 to 3 months of age, 6 to 8 or 6 to 10 adult, and then old, usually 20, 22, we don't go over than that, but sometimes 24, and older than 28, they are geriatric. And the question we had was to understand what changes with aging within these stem cells, focusing on these two old age periods. Because if we understand the causes that underlie the regenerative decline, we'll be able to do. So these two aims are basically what we had in mind over 10 years ago, and they still are the main questions that we have in the lab. Try to understand the causes or the regenerative decline and try to do something to halt them or reverse them. So this is a typical example of an injury to the skeletal muscle using a myotoxin, cardiotoxin, and as you can see, both the number of, so the size of each fiber is reduced in all mice, and it's reduced in geriatric mice. But on top of that, the geriatric mice have reduced formation of fibers. I don't want to say that all mice 28 months of age, I mean, we do have variants. It's not easy to get 28-old mouse, month-old mice, 28 months, sorry. But what I mean is that what we found more striking from these experiments that we've seen all over time is that despite the variability with mice, we always observed a sharp decline just 4 or 5 months older in geriatric mice compared to old mice. And we never had the chance, we never could do parabiosis experiments, but we did these transplantation experiments from donor satellite cells from adult or geriatric mice and transplant them in a recipient mouse of the same age. And we could see that the geriatric mice always did worse, not compared to the adult, but compared to the old. So in addition to the external environment, there are changes in the geriatric satellite cells that, at least in transplantation experiments, cannot be overcome by a young environment. So this is something that I will take a bit more careful now, based on a question that I think it was by Lucia. So I think that we observed in bulk RNA sequencing or microarray analysis that satellite cells from mice at geriatric age, but not at young age, increase the levels of P16. That doesn't mean we didn't have a single cell then. Now that we have single cell, we can see that not every cell has more P16, but definitely there is more P16 in very old geriatric mice than in just old but not geriatric mice. So, and these cells are pre-senescence. They don't have features of senescence, even if they are in the geriatric muscle. They are just, they are more resilient or more resistant to activate. So when you do an injury, these cells will take much longer time or will not activate at all because they express P16. But this is a subset of cells, not all the cells. But in general, they activate worse than the old cells. And among other many explanations for this behavior, we think that those that express P16 have more chances that after an injury to undergo senescence than those that don't express P16. So how do they become pre-senescent, those cells that express P16? So we did bioinformatics analysis from transcriptomic data of quiescence and activated satellite cells, or young and old, and we came with autophagy as a prevalent prevalent, or proteostasis as a process that is characteristic of quiescent cells, probably having processes that control that take care of the intracellular toxicity of the cell, confers, well, it's important for the cell to maintain its stemness. So what we wanted to see at that time, why a quiescent cell would need this autophagic activity. So we used a LC3 GFP mouse. In this case, the GFP protein is linked to LC3, which decorates the protein that decorates the inner and outer membrane of the autophagosome. The autophagosome is the double vesicle that is formed to engulf proteins or organelles that need to be degraded into the lysosome through the process of autophagy. So this is a fixed picture of puncta of GFP using these GFP LC3 mice. And you could see here that old mice, satellite cells freshly isolated from old mice, have more puncta and more intense than the young satellite cells. But this is not, that autophagy is a dynamic process, and this is not a dynamic process. It's just a fixed picture. So we did very classical flux experiments in which we briefly, after sorting, we briefly incubated the cells with baphyllomycin to block the elimination of autophagosomes into the lysosome. And as you can see here now, the young satellite cells incubated in the presence of baphyllomycin have more puncta compared to the old that don't have more puncta. And if we look at the mean fluorescence intensity of the GFP, we can see a small but always very reproducible shift in the intensity of GFP in the presence of baphyllomycin in the young but never in the old. So that indicates that there is basal autophagy in the young and it declines with aging. To make a long story short, because this was published, what we could see is that autophagy starts to decline with aging, and what we found indeed was that it was mitophagy that was declining with aging, and we found accumulation of non-functional mitochondria, which leads to the accumulation of reactive oxygen species, and this epigenetically de-repressed P16 in those, and that led to the expression of P16 in some cells that are those that we have data to show that are the ones that will undergo senescence. So can that be reversed? So I mean, this block. So this is not a reversal of senescence to my knowledge, even in there are groups suggesting that senescence. For now, let's consider it irreversible. What we can do is to prevent the entrance into senescence. So what we did here is to treat mice for 2 weeks with rapamycin, and then we obtained satellite cells from treated or not treated mice, or did an injury to the treated or not treated mice, so but not treating rapamycin during the injury. That would block proliferation. So what we saw is that we could reduce autophagy in satellite cells. This is a flux experiment in mice treated in vivo with rapamycin. And transplantation experiments, this is young and controlled satellite geriatric cells. You can see that the young transplant better if they come from mouse treated with rapamycin or from satellite cells ex vivo treated with infected with an ATG7 or ATG12 or ATG5. They proliferate better in vivo and also in vitro. These similar results have been obtained also by very nice work by Emmanuel Passeguet in hematopoietic stem cells. So I promise that I always have this slide, so it's not because Tom Rando is here. So I would like to show that we think there is not just autophagy, maybe as Ana Maria Cuervo is proposing with chaperone-mediated autophagy, other types of proteostatic pathways are not working well with aging, and we will have proteotoxicity. And this will lead to a pre-senescence state, and if there is an injury, we might have subsets of cells more injured that will go into senescence. And this can be prevented, I don't think reversed, by what we showed is rapamycin, which is a pharmacological reinduction of autophagy. We also showed by antioxidants reducing the levels of ROS in the satellite cells, or genetically. And of course, other labs like Tom has shown the effects of sharing the blood within two mice, how the rejuvenation effect, exercise, and others have also worked on caloric restriction, including Amy Wagers or others. So, but what I wanted also to tell you that the satellite cells know what time of the day it is. So, this is a transcriptome of satellite cells sorted every 4 hours for 6 time points during 24 hours. And we did, those are mice that are 5 months of age, adult mice. And these are, we just look at a bioinformatic package, the GTK cycle, and this indicates which genes have an oscillatory pattern, or if they are flat, if they don't oscillate, this algorithm would not tell us. Well, we always tells us which genes are rhythmic. So in this analysis, we observed around 1900 genes that were rhythmic in adult satellite cells. When we did the same with mice that were not just adult 5, but in parallel 2 years of age, and did the same experiment, we found that another number of genes, and very importantly, we saw even more genes that were circadian in the old. Interestingly, 28% of the genes that were circadian in the old continued to be circadian in the old. 72% of the genes are only circadian in the adult, and what was more interesting was that around 1600 genes were circadian de novo. So it indicating that there is a rewiring or a reprogramming of the transcription gene expression, circadian gene expression program. And it was always thought that the circadian rhythms are dampened with aging. Well, at least for epidermal stem cells and muscle stem cells, and now we know with skeletal muscle, there is no damping of the core clock machinery. This is published. There is just a new program being expressed, and even more abundant. And so, for example, what we could see is that autophagy is circadian only in adult, but many inflammatory genes, not just they get expressed, but get expressed in a circadian manner only in all satellite cells, and also stress cells genes. And we could discuss or speculate why this, and this is something we're trying to understand. So with caloric restriction, a long-term caloric restriction for 5 months, we could bring back, well, not bring back, prevent the rewiring or reprogramming of this old transcription program in the old satellite cells to be more similar. So this is old satellite cells from not diet, so ad libitum fed mice or caloric restricted mice. Now we could bring back these genes are very similar to the young circadian rhythms. So what we say is that the satellite cells do some of their homeostatic functions at different times during the day, and this includes autophagy. During aging, these functions become more inefficient, some of the homeostatic functions become not circadian, and now some functions related to stress become circadian. How long do I have?
No, don't worry.
Okay, thank you. So I wanted also to discuss about satellite cells. So our classical work was, we were talking about satellite cells, but we wanted to, there was in the literature many well, not many, but at least several groups had already reported that satellite cells were heterogeneous. And these studies were based on transgenesis, using beta-gal reporters, using labeling studies. So we knew that satellite cells were not alike, all of them. And so we wanted, a few years ago, we started to see if we could study muscle stem cell heterogeneity or diversity in the absence of transgenesis or labeling disease. We get a mouse, and doesn't matter if it's from a collaborator, to study satellite cell subsets. And if this was possible, to study this heterogeneity over time and the mechanisms that would regulate this heterogeneity. So this is a typical one of the few sorting protocols for satellite cells with alpha-7 integrin CD34, and you can see that CD34 is expressed in quiescent satellite cells from basal or resting muscle, and its expression goes down 3 days after cardiotoxin injury. But Laura Garcia, that some of you may know, she was one of the best people, students in my lab. She was always noticing, she loved to do cell flow cytometry, was saying, there is not a concentrated population of CD34 positive cells. There's always an elongated and elliptical shape. So she thought that maybe we wouldn't have discrete subpopulation of satellite cells because they are a continuum, they're quiescent satellite cells, but there might be discrete differences, and to prove that, she sorted the 15 higher CD34 high and the 15 lower, the 15% CD34 low, and see if she could distinguish subsets or subpopulation of that whole population of satellite cells. And molecularly, after transcriptomic analysis, she could see that there were, this is quite stringent, that there were genes that are unique for the CD34 high and genes that are unique to the CD34 low. And among them, in the CD34 low, you could see myogenin transcript, and also other P57, you could see more, some myogenesis related genes or differentiation genes. And here we could see notch genes and other genes that are not represented, but more stem-like related genes. When we looked at bioinformatically function, we could see that the CD34 high satellite cells had much lower activities than the CD34 low. The CD34 low, even if they are quiescent, at this level of bioinformatic analysis, they had more machinery of transcription, mitochondria, OXPHOS, muscle differentiation compared to the CD34 high that was very difficult to even get functions associated. So we could see that CD34 was reporting on molecular differences. What about functional diversity? So what Laura did was analysis in vitro and in vivo. So she could see that in vitro, the CD34 high had more clonogenic capacity and more self-renewal capacity doing reverse population analysis. And this is not black and white, but always the CD34 high gave more fibers upon engraftment than the CD34 low. So based on molecular and functional characteristics, we thought that the CD34 high would represent a more genuine, and now we could say maybe more dormant state than the CD34 low, which is more primed for myogenesis. And we call them CD34 genuine and primed. So that this diversity changed over time. So Laura took satellite cells, alpha-7 integrin CD34 from mice that at 8 days of age after birth, 3 weeks, young, old, or geriatric. And she did I won't show you the results of the neonatal and young, but or juvenile, but this is the transcriptome that I showed you before at young age. This very more active pathways bioinformatically in the CD34 low than the CD34 high. And the question is, does this transcriptome, these functions change with aging when we get CD34 high and low from old mice and geriatric mice? So these signatures are very reproducible in satellite cells from all mice. We get less cells because you lose half of the cells, but those that we get, transcriptomically, they're very similar. But at geriatric age, they're not very similar, they're quite distinct. And this is the pathway enrichment analysis of the CD34 high geriatric satellite cells. They resemble more the CD34 low of the young or of the low or of the old. So they're kind of more active, and we think that's why we lose them. So they're not, CD34 allows us to isolate the cells, but they are not as stem as the CD30 as the young or the old. So do we see any difference in functionally within this CD34 high and low, or genuine and primed at the different ages? So this is transplantation experiments of the CD34 high or genuine from mice that are young, old, or geriatric. And we can see that the geriatric do much worse than the old or the young. And while the prime do always bad, regardless of the age. They never do well. What regulates this diversity? Well, we tried to do computational analysis, and we found that there was a niche-derived signal, IGF-1, which would impact on the receptor on the satellite cell, and by activating AKT would repress FOXO in the genuine satellite cell compared to the primed. So we postulated that if we inhibited AKT by activating FOXO, forcing it to move into the nucleus, we may reinforce the genuine cell state and convert a primed into a genuine, or a geriatric into a younger cell. So this is just to show you the need to have FOXO to have regeneration, FOXO 1, 3, and 4. When we delete the 3 FOXOs, we lose all the CD34 high satellite cells, and we don't have good regeneration. But if we silence, if we silence myogenin, we get somehow a recovery. So if we block the master regulator of myogenesis, these cells are preventing from entering the precocious differentiation, even if they're lacking FOXOs. So this is the model that we propose. So at neonatal age, most cells are proliferating, and they have not decided yet their fate. By 3 weeks of age, they start here represented this more bluish color and this more yellowish color. So when they are young, we have two discrete, in a continuum, two discrete populations, although we have a medium population as well. The more stem cells are maintained by high FOXO in the nucleus and low AKT signaling. And this subpopulation of, even if they are less, this is maintained up to old age. And the primed satellite cells, this subset is always low in FOXO in the nucleus and has high AKT. While at geriatric age, this FOXO in the nucleus starts to go down, we have more AKT signaling, and this population is molecularly more similar to the primed, so they're closer. So, well, I think I'll leave it there. It's just, so I would like to think that we could think of rejuvenating strategies in which we can think of promoting the most fit, genuine or superior stem cells, as if we could try to endogenously understand the regulators of these cells that resist aging up to quite so the heterogeneity of the cells needs to be exploited, leveraged to see how to promote those more resistant or fit stem cells. And I want to thank the collaborators. This work was initiated, most of it, and done by Pedro Sousa and Laura Garcia. And those are very good collaborators I have in the lab, Joan Iserme, Eusebio Perdiguero, and Antonio Serrano. And for some of the work, I collaborated with Vittorio Sartorelli, Antonio Musaro, Marco Sandri, and which are known to all of you. Thank you.
Thank you, Gura. Fantastic work. And now we can open the discussion, please. There is a question. Can you come here? No. In streaming, I cannot hear you otherwise. No. That microphone doesn't work so much.
I have a very naive question. In your geriatric population, do you see a sex bias? Do you have more females than males? And did you ever look at that?
We were mainly using male. We have never looked at the females versus
patients,
male.
we see the difference that a male, female, like patients do different.
There are, I know there are now differences in the TOR signaling, which might be very important for this, at least for the P16 expressing cells, but we have not studied that, to tell you the truth.
Carles?
Okay. I try to speak, and then if not, you can speak for the people online. So thank you, Gura, for your presentation. I like a lot your speech about diversity and heterogeneity, because it's also in my interest. And you speak about the diversity on the same population. But I want to add another layer. What about the diversity along the body? So we know that the process, the senescence, or even we learn from the disease that not all the muscles or not all the body degenerate at the same level or the same paces. No? So, for example, in the regeneration, it's not the same the regeneration of the maxillar than the regeneration of the tibialis. They have different capacities. So, my question is, have you ever seen about or look about this senescence process in different muscles in the body? And if you another thing, and if you think it should be a good control to this experiment to use the different muscles of the body. So not to use, for example, to use a tibialis versus a maxillar, muscles that differentiate, degenerate.
So maybe I should re-emphasize that we don't know senescence in the absence of injury. It doesn't matter if it's young or if it's geriatric. I had one slide only that I eliminated, and it's a pity. Maybe you see one or two senescent cells. Maybe in humans you see a few more because the chronological age is longer. So we only see senescent cells, satellite cells as a population, and basically senescent cells from other types of, coming from other niche cells, only after an injury. So I think if the injury is strong enough, at least in all limb muscles we have seen it. It doesn't matter if it's gastrocnemius, if it's the plantaris, because we've done all of those. I haven't done, for example, we haven't done senescence in injury in extraocular muscles, and based on the work of Brenda Komai and in Sharagin Tahba's lab, showing that the extraocular satellite cells might be more resilient, or at least 10% of them seem to be more resilient to damage and be more stem. Who knows if the process of senescence will be reduced in those cells. But yeah, I mean, we have systematically we have only analyzed the limb muscles after an injury, and of course also in MDX mice there are many senescent cells. Again, not only satellite cells, from many other cell types.
Thomas?
Yeah.
Yeah, if you can come here, it's better.
No, I want with this one.
So that was beautiful, and I love your examination of diversity. You've done this more than others, but others have shown subpopulations like the Tajbash PAC7 high, PAC7 low, and Rudnicki MIF5 low. Do you know if there's a relationship between your subpopulations and those? Because I think you're the only ones who really looked at this with age. I just wonder if that's a...
Yeah. So when I've talked a lot with Sharagim about this, when we use the PAC7 NGFP reporter from Sharagim, we don't see a coincidence in the CD34 high with the PAC7 high completely. That's it. And and we've never used the Mi5 beta gal reporter. Yeah, so, but when we look at the genes expressed in the CD34 high, we see the notch receptor, we see notch, we see markers that are reported by other groups to define the more stem or more dormant populations. But with the reporters, we have not, which is something that bothers me a bit, but thank you.
Aurelio?
So we were dreaming with Thomas to rejuvenate. I want to ask you, can we use your, let's say, triggering pathway for make the cells senescent in a rhabdomyosarcoma? I will never look, because as you know, one of the markers that you mentioned, myogeny, is indeed a marker of rhabdomyosarcoma. So can we use this kind of senescence and get rid of this tumor?
But myogenin was here expressed in the CD34 low cells, and not in the senescent cells. But I think maybe I missed the question, Maurilio. So,
because rhabdomyosarcoma is a kind of juvenile myoblast that stay quite active without terminating differentiation. Can we force senescence in this tumor?
Can we force? One could force senescence in those cells, but if you force senescence, you also maybe lose the progeny unless you find a specific way to just induce in those cells that are P16. Yeah, that's a good idea. Yeah, I mean, all I know is if we took, that was an old experiment in our first senescence paper, if we took young satellite cells and infected them with a P16 expressing construct, these cells in culture would become senescent by higher doses of P16. Yeah, one could do that, well, I guess. Yeah.
Yeah, Andrea, please.
So, maybe I will reply
later. I was just curious, as an expert of senescence, what's your definition of presenescence? And because I mean I work on senescence and cancer, but I don't know about muscular. The second thing is, do you have an idea of some senolytics that work behind nabitoclax treatment? So are you guys in the muscle field studying or screening for different senolytics?
Yes.
Where so is tissue specific?
As we are at home here, sorry if I. So what I mean by presenescence is a cell that, so the satellite cell, subsets of the cells at geriatric age of the satellite cells and other cell types express P16, they have some DNA damage, but of all these characteristics to that, because there's no universal marker of senescence. So of all those things there, maybe one or two are only present in.
So they don't have the SASP?
No.
They don't resist to apoptosis, for instance?
No, they're quiescent. Yeah. They're quiescent, but they don't have. I mean, they're not senescence because they don't even. So you know that for senescence you need a proliferative stimulus. Otherwise you will not have. So it's almost impossible to have a senescent cells in a tissue that is postmitotic, that doesn't have proliferation. You will get only senescence when you injure it, right? So if you go here.
Okay, so the definition is a few markers versus many markers.
Yeah. Okay. So here is a geriatric muscle basal. And this is beta-gal after injury. Yeah. You don't have it when you don't have the injury stress. You can have also senescent cells, and those cells are not just satellite cells, there are many other cell types there. If you look at young muscle, you will have also senescent cells, you just have less, and they are and they don't remain so much. Probably this is nothing to do with senescence, but because of failure of the immune system.
But the senolytics.
Senolytics.
They're going to work on the full senescent cells, not on
quercetin and azatinib. You treat them from day 3 to day 6, 4 days, and measure, for example, embryonic myosin heavy chain. So in our hands, we never eliminate them. We only reduce them to maximum 40 or 50%. Then for each fiber, you get an increase in the size. And if you wait longer, you can also get an increase in force. You can do it genetically also if you use the P16-3MR mice.
Did you use MCL1 inhibitors, for instance?
I use 13 senolytics and alone or in combination. And this is the one that works better.
Okay.
No, ABT263 also works. Well, navitoclax. There's one every week, basically, but those are the ones that work better in our hands. But better means 50% reduction, not more, actually.
So, Pura, I have a curiosity, actually, related to the Carles question. During aging, the fibers that are most sensitive to damage or anyway that are lost preferentially are the fast fibers. And actually there is basically no major insight into the role, into the mechanism that is involved in this process. But if I will remember, the slow fibers contain more satellite cells. Do you think that the satellite cells isolated from the slow fibers compared to the one isolated for the fast express or activate this specific pathway such as FOXO to maintain the genuinity?
Yeah, that's something that I wish we could be continuing doing, but unfortunately when Laura left, I think, so when we look at CD34 positive cells, they're brighter in the, so CD34 is more high in the oxidative muscles than in the high muscles, like if closer to oxygen or endothelial cells, they would be even more stem. I don't know, and maybe they are, that's why we lose them less there. But this is all pure speculation. And you're welcome to work on it because we're not working on it. Yeah, but I'm sure there are satellite cells like other groups. We're not looking into that satellite cell diversity intermuscle, and not just maybe the extraocular versus the limb, but also among the limb muscles. We just have not looked carefully enough.
Okay, there are questions from the audience. Jacopo, can you check please if there are on the chat?
No questions.
No questions? Other questions from the audience here in presence? Okay, if not, thank you again, Pura. And Andrea, please. your microphone. The microphone is yours to introduce the next speaker.
So it's a pleasure for me and for the IPSA Foundation Scientific Advisory Board to introduce Feliciano Protasi, which is a full professor of physiology at the University of Gabriele D'Annunzio in Chieti. So Feliciano is running a lab of more than 10 people working actively in the field of aging and myopathy, in particular with a focus on calcium metabolism. So we're really happy to have you here. So the podium is yours. Thank you.
Thank you very much for this introduction. I would like to start thanking, first of all, Antonio Musarò for this invitation, and then the Accademia Medica of Rome. And it's a great pleasure for me to be here. And I want to go back to the time in which Antonio invited me for this talk. And the title at the time was slightly different. It was a preliminary title, and the title was Understanding Muscle Aging and Identifying Ways to Induce Tissue Rejuvenation. And I think my presentation today probably fits better this title than the new one, because today I will talk about the remodeling of muscle fibers as a consequence of aging, as a consequence of inactivity, and also the effect of exercise on this muscle remodeling. And the main interest of my laboratory since I came back from United States in Italy, it was 2002, is to study the sarcotubular system of muscle fibers. And the sarcotubular system is a complex system of membranes that here you see in blue and in yellow, which are in internal membranes that are kind of wrapped around each single myofibril. And the myofibrils are the structures that allows muscle contraction and shortening. And the sarcotubular system is formed by two independent system of membranes. The one that here you see in blue, which is the sarcotubular, the sarcoplasmic reticulum, which contains the calcium that is needed to activate muscle contraction. And these are intracellular membranes. They are actually the specialized system of muscle fibers that in other cells is the endoplasmic reticulum. And then you have these yellow membranes here, which is the transverse tubules, which are, they're not really internal membranes. Actually, they are invagination of the surface membrane, which carries the action potential inside the muscle fibers. And this system is the system that communicating with the sarcoplasmic reticulum allows the release of calcium to activate muscle contraction. So how did I develop this interest into the sarcotubular system? Then, about a few months ago, I was preparing a talk for my institution, and I went back and looked at the title, sorry for the Italian, the title of my thesis in biology, and already back then in my title there was reticulosarcoplasmatico, that is sarcoplasmic reticulum, and there was also ryanodine receptor, which is the ryanodine receptor is the calcium release channel of the sarcoplasmic reticulum. This is the reason number one why my lab has interest in studying this system of membranes. The other reason is that when after the thesis I moved to the United States to the laboratory of Clara Franzini Armstrong, she was an expert in studying this system of membranes, and she was using actually electron microscopy, and this picture is a picture taken in her lab in which you see the sarcotubular system stained with Golgi staining, and you see these membranes are they are dark, and also if you see here in yellow now I have false labeled the transverse tubules that are the system that carries the action potential inside the fiber. And this, the communication in between transvertebral and sarcoplasmic reticulum occurs in specialized intracellular junctions that you see here are formed by 3 elements. And in another type of preparation, again in electron microscopy, you can see this structure that are called triads that are formed by a transvertebral, that here you see this is kind of flat, and the sarcoplasmic, the terminal cisternae of the sarcoplasmic reticulum on both sides. And these are named triads because they are formed by 3 elements. And the triads are the sites of excitation contraction coupling. EC coupling is a mechanism that allows the transduction of the electrical signal, and here in this cartoon you see the electrical signal that goes into the transvertebrals. Here there are proteins that act as voltage sensors, which senses this change in polarization of the membrane, communicates with another protein on the sarcoplasmic reticulum, which is the ryanodine receptor, the calcium release channel. Then after this communication, the calcium is released, and this activates muscle contraction. And muscle contraction stops when the calcium is brought back into the sarcoplasmic reticulum by another set of proteins, which are the SERCA pumps, sarcoendoplasmic reticulum calcium ATPases. And obviously, calcium handling in skeletal muscle fibers is more than just EC coupling. So EC coupling, I told you, allows release of calcium from the sarcoplasmic reticulum. The SERCA pumps brings back the calcium, but there are many other mechanisms that modulate the levels of calcium inside the skeletal muscle fibers. In the cytoplasm, there are many calcium binding proteins. Today we know also that in muscle, also the entry of calcium from the extracellular space, has something to do with the resistance of muscle to fatigue. But today I want to focus on another mechanism. So some of the calcium that is released during EC coupling actually enters the mitochondria. And this entry of calcium into the mitochondria is very, very important to activate aerobic production of ATP. And this makes sense because you have to think that when muscle is resting, we don't need much ATP. But when we start to exercise, for example, our organism starts to use more oxygen, and the oxygen is used by the mitochondria, and this entry of calcium into the mitochondria acts as a signal to tell mitochondria to start producing more ATP during the respiration, aerobic respiration of these fibers. So today the main focus of my presentation is the interaction in between the mitochondria and the sites of calcium release, which I told you before, they are named triads. If we look, we know very well that skeletal muscle fibers are plurinucleated cells that have a very, very highly organized intracellular space. This intracellular organization is due to the cross-striation of the fibers. So you have many myofibers which are aligned with one another, and this creates a cross-striation that gives to the fiber a very, very orderly intracellular organization. However, when we look at images of cartoons of skeletal fibers in textbooks, what I notice is that many times these organelles here, which are called mitochondria, they are not put in the cartoon in a specific position. Is this really the situation in skeletal muscle fibers or not? And this is why we started to look using electron microscopies. This is the technique I learned in Clara Franzini-Armstrong laboratory. When we look at the position of mitochondria inside skeletal muscle fibers, well, this is not the position you see in the cartoons in the book. Actually, also mitochondria, they occupy a very specific position. And here I labeled in with these arrows, these structures here, which are the triads, the structure that releases calcium, and the mitochondria are specifically placed next to the triads. And now if I enlarge one of these details, what you see a triad, this is the site of calcium release, and the mitochondria are placed just next to the sites of calcium release. So is this true in every type of fiber? Antonio before was talking about slow twitch fibers, fast twitch fibers. In slow twitch fibers, we also have another population of mitochondria, which is more longitudinal between myofibrils. But also in slow twitch fibers, you have these mitochondria that are placed next to their calcium release sites. So does this structure have a functional meaning? Well, we think yes. In a study that we published in 2009, here you see a tomography. You are looking at a thick section of muscle, which while is rotating, and we did the tridimensional reconstruction of this couple between one triad and one mitochondria that you see here. And I apologize, the movie is not working. This movie in yellow and green should rotate, but it gives you the feeling. We described in this paper in 2009 the fact that the triads are actually linked specifically to the mitochondria that you see in green, but these little red proteins that at the time we called tethers. Still these days we are not sure what is the molecular identity of these tethers, but back then we started to hypothesize what is the role of these little linkers that keeps the mitochondria next to the sites of calcium release. And now I want to show you this movie in which you see this green labeling. This movie is about 20 minutes long, and this is our mitochondria stained with mitotracker green. This is not muscle. These are HEC cells. So if you look at the position of mitochondria inside HEC cells, in 20 minutes they move a lot. And now these days we know the mitochondria undergoes fusion and fission, and they're very dynamic inside the muscle fibers. But is this true also in skeletal muscle fibers? So when we look at skeletal muscle fibers, and now here you have a movie of skeletal muscle fiber, you see the mitochondria in green, they make this beautiful double cross striation across the fiber. Well, in 20 minutes, the mitochondria in muscle fibers are not as mobile as in the other cells. So, and back then we did also in this paper some experiment using an hypotonic solution trying to detach the sarcoplasmic reticulum from the mitochondria, and actually we found that this tether kind of stretch. So we hypothesized that maybe these tethers are important for holding the mitochondria in a very specific position next to the calcium release site, and maybe this is important functionally. So probably we know that muscle is the more metabolic organ of the body. So muscle can change the oxygen consumption and the ATP production from rest to exercise for many times. Maybe the mitochondria need to be next to the release site to be metabolically efficient. So we developed this idea, and at the time we were working in other projects, studying aging, studying denervation, and doing other project, we were actually collecting some information that was important for this idea, because when we were looking at muscle, denervated muscle, here you are looking at the muscle denervated in rabbits, 10 weeks, and you see that if we denervate muscle, so we have a muscle with no activity at all, no innervation, then the mitochondria lose their position, their specific position. Actually, you get areas like this, in which the mitochondria are completely missing, and the mitochondria have migrated longitudinally. So in this position, they are no more next to the sites of calcium release. This is inactive muscle. Have we seen this only in rabbits? No, we've done the same experiment in muscle of rats, in muscle of mice, and also in humans. So if you inactivate the muscle, the mitochondria lose their specific positions next to the sites of calcium release. So then we started to study aging. We have also our center where we have our laboratory was the Center for Studies in Aging, so there was an interest in aging. We started to look at aging. These are muscle of mice of 2 years of age to reconnect with the talk of Pura. This is our old mice at 24 months of age. And we noticed that in these mice, a percentage of mitochondria was detaching from the triads. So they were losing proper position, like in denervation, not this many mitochondria, some of them, they were losing. And down here, there is a quantitative analysis of the number of mitochondria that lose position next to the triad. So during sedentary aging, these were mice that were living in normal cages with no possibility of exercising really. So the mitochondria were losing proper association to the sites of calcium release. In the same paper, we also did some studies of oxidative stress, and we proposed that these changes that occur with aging was due to oxidative stress, and probably mitochondria that are not very active, they produce too much oxidative stress, damage to the membranes, and also probably disruption of the tethers, and the mitochondria were getting away from the sites of calcium release. So then obviously there was a new question that was arising. Could the association between mitochondria and triads, which is challenged by aging, be maintained and improved by exercise? And we did this study first in collecting human biopsies in collaboration with Sandra Zampieri, Antonio Musarò, and Helmut Kern in Vienna. We were collecting muscle biopsies from different groups of people. These are humans, young adults, age 26 years of age, senior sedentary, these were people that were not doing exercise, of 70 years of age, and then serial sportsmen. These were people that were doing regular exercise for 3-4 times a week since their midlife, since 40 years of age. So they were doing regular exercise for a long time. And we published this paper, it was 2015, but I want to show you one picture from this paper. On the left, you see EM images of muscle fibers from sedentary seniors. And on the other side, these are the muscle of sportsmen. And now you can see that here, if you look at the structure and orientation of the triads, is very disrupted. Many mitochondria are missing. You have a lot of accumulation of these granules of glycogen. But on the other side, when you look at the muscle of people of the same age, but they regularly exercise, now you don't have this accumulation of glycogens. And the mitochondria that here are indicated by the white arrows are in the specific position that I've described to you before. And down here you see they are coupled correctly next to the sites, the triads of calcium release. And we have repeated, since in humans we have little pieces of muscle and we can do very little with this muscle, we repeated the experiment in mice, 2 years old mice. We did the experiment also of fluorescence. Here you see in green the staining of mitochondria in aged mice, and down here the staining of mitochondria in mice that were trained in cages for voluntary running. These were mice of 2 years of age, but in the second part of their life, from 12 to 24 months of age, they had access to wheels for doing voluntary running. They were not forced to run. So the mice tend to run at night, and they run quite a bit of kilometers a night. And I think our mice were running from 1 to 2 years of age. Obviously, the amount of activity was going down with age, but they were keep running. And now their mitochondria, after aging, they are not misplaced like the mitochondria in sedentary aging, but they have this correct position next to the release sites that here are labeled in red. This is the staining of the RNOD receptor, of the calcium release channel of the sarcoplasmic reticulum that I mentioned before. So in this study, I'm not showing you the function, but good positioning of mitochondria equal better resistance to aerobic exercise and more strength and everything. So last part of the story, very short, and this is still unpublished. These are experiments we are doing now in collaboration actually also with Antonio. So loss of proper position of mitochondria occurs only in in aged muscle. Here there is a mistake, in aged muscle, as a consequence. The question is, we have seen this in aging, but this loss of position of mitochondria, would occur also in adult muscle as a consequence of inactivity? And is this process reversible? So with aging we get this. Can we get this misplacement of mitochondria at the end of triads also in adult muscle if we keep the muscle inactive? And is this process reversible? And what we are doing here is we are immobilizing one leg of the animal, only one, of the hind limbs for only 6 days. And then after this unilateral immobilization of the hind limb, what we did is that we did, after 24 hours of this immobilization for 6 days, we did 2 weeks of retraining. In the first week we did training on the treadmill. This was forced running on the treadmill for only 30 minutes, 4 times a week, and the speed was very low, 5 meters per minute. And then in the following weeks we did 3 times of training at a higher speed of 10 meters per minute. So, and the preliminary data we are collecting are in this image. I try to be here. We are looking at the number of calcium release units. These are the triads, which with immobilization the number goes down, and after rehabilitation, retraining, the number goes up. Number of mitochondria, also the mitochondria content goes down and goes up with retraining. And now here you have the analysis of how many times the mitochondria is correctly placed next to the triad, and we see that we lose association very quickly, and then if we retrain the muscle, we can get back the mitochondria in the right position. So this is the final slide to summarize what I've showed you, so that sedentary aging causes uncoupling of mitochondria from the sites of calcium release. This we have seen also in several myopathies, and we have seen this also in denervation. But then regular exercise during life is very effective in maintaining the mitochondria in the correct position. It's not only matter of position, it's also matter of number, volume of mitochondria, and number of calcium release units. But what we are seeing lately, and this is still a paper that is not published, is that short-term inactivity does the same thing also in adult muscle. But the good news is that if we retrain, the mitochondria are quite plastic, and they can get back to the normal position. So today I've showed you also only the structure, but in our papers there is also the function. So the correct structure is always the base for a proper function of muscle. And in a separate project, I don't have time to present data there, but we are studying the remodeling of the transvertebral and the sarcoplasmic reticulum in response to exercise. And in that project we are studying how the membranes remodel to allow calcium entry from the extracellular space. But I want to tell you that there we are starting to study a little bit the molecular mechanism underlying this remodeling. And we have found that every one of you knows that during exercise the temperature of muscle goes slightly up, and also the pH goes slightly down. So we are studying remodeling of muscle, not in the mouse, but dissecting the muscle outside the mouse and doing fatigue protocols. We get the remodeling of the membranes, and we are studying separately the effect of temperature and pH, and it seems that pH and temperature have a lot of influence on the remodeling of these fibers. So then I want to conclude by thanking the people in my lab that did these studies during these 10 years. And my collaborators, Helmut Kern, I mentioned before, and Antonio and Sandra Zampieri in this project, and obviously all of you for being here. Thank you very much.
Thank you, Antonio, for this beautiful talk. There are questions? Yeah, there is a question. I'll try to read it.
Did you try to make some electrostimulation in the mobility muscle?
Oh, that's a very good question. When you say this, I kind of think that maybe you have read some of our papers. When we were looking at the denervated muscle, this project that we were doing earlier, we have done electrical stimulation in humans and in rabbits, and we have looked at this and the electrical, and also with Antonio, we look at the effect of electrical stimulation in aging, and we found that electrical stimulation is effective in restoring this alteration that we get with denervation in activity and aging. Yes.
Other question, Antonio?
Feliciano, curiosity, do you think that in all of these experimental model you can observe some of the pathway that Tom Pura described during aging? So the physical exercise that work on the mitochondrial behavior and activity. Do you think that also involve some of the pathway like autophagy?
So the question is if you see autophagy in your experiment, just for the people.
Well, some of the molecular pathways, we do, as you know, that we don't have the expertise to look at this, but in collaboration with also with you in the past, we have looked at some of the molecular markers that in aging, and I think that those pathways for sure they have to do with this remodeling. I also think that we should look, actually, this is something that we are starting, we should look at some of the pathways that control the remodeling of the mitochondria, and we are going to start to do this because we know that you have some pathways that controls fusion and fission of mitochondria, and we should look at this, yes, definitely.
Another question?
I also...
Do you want to?
So I'm curious because you said that you see the same mislocation in myopathy, right? So I'm wondering if in the case of chronic inflammation or chronic degeneration, you still have a completely mislocation of triads with mitochondria. So did you analyze this phenomenon in dystrophic fibers or other form of myopathies?
No, I didn't look at those models, no dystrophy, no, you talk about inflammation, no. But I've seen this in other myopathies like central core disease or malignant hyperthermia, because our interest is mainly in those myopathies that are caused by mishandling of calcium. So I've looked in those and I've seen similar phenomena. Also sometimes when there is excessive calcium leak from the sarcoplasmic reticulum, we see the mitochondria getting damaged for calcium overloading, and but not in those cases. Obviously, I will be very interested if you have samples to look at it.
Pura, you have a question? Can you come here because it's very difficult for me to repeat the question. Very complex question.
Yeah. So, do you have any idea why the mitochondria get detached? No, from with aging or immobility, not really.
Well, I've showed you in one slide that we think that maybe oxidative stress has something to do with the damage of membranes and probably also of these proteins, the link, but yeah.
Maybe I looked at it, I'm not used to electron microscopy, but were your mitochondria very large and elongated, right? So that could be two things, or the fission is not working, or the fusion is hyper working, or autophagy is not working. So they're not degraded, any of the three?
More than they get bigger, I think that they change position, because three-dimensionally, the mitochondria are wrapped around the myofibrils, like when we cut with the electron microscopy, we see them round, but they're not really round. They are kind of wrapped around the myofibrils, and I think that when they change position, they move from here to along the myofibril. So, because when we measure the volume, the total volume of the fiber occupied by the mitochondria is reduced. So, but definitely we should look at fusion and fission proteins. This is true, but they don't get bigger, they just move away from this transversal position and they become longitudinal.
But what about if it's vice versa, that because they change their shape, they don't fit where they should be?
It's possible. I cannot We have looked also at another model during development, for example. During development, we have many more of those longitudinal mitochondria that are not yet in the right position. So, but definitely it could be, yes.
There is another question.
Nice talk. I am an endocrinologist, so I was wondering whether thyroid hormone, which is a very important regulator of mitochondrial function and also calcium intake, the SERCA1 is a well-known thyroid hormone targets. Do you know whether in thyroid disease with the concomitant alteration in thyroid hormone level there is any kind of misposition of this mitochondria in the myofiber?
Yeah, I've never looked at such a model, but definitely it would be very good idea to look at it. Yes, though I don't know.
So there is another question, if you can come.
No, yeah. But I can repeat the question.
Connecting to the question of Professor Sampolesi, have you seen the mislocation also in pathologies like progeria, for example, or laminopathic phenotype? Because this is, or maybe could it be a good model for pathological aging? Because you study your effect in natural aging of mice or the people.
Okay, so
progeria, you have
so the question is if in progeria or in other cases of pathological aging do you see also this mislocalization of mitochondria?
The question is, unfortunately, is no. I never looked at those samples, and definitely if somebody has the interest to have this kind of answer, I would be happy to analyze samples. Yes, no, never.
Okay, so if there are no further questions, I will invite Antonio to introduce the last speaker and to give the conclusive remark.
Yeah, the last but not least speaker is Professor Maurilio Sampolesi. So Maurilio got a PhD in cardiac pathophysiology at the University of Tor Vergata, and then he did a lot of actually postdoc position, one at the Center Research Institute in Osaka, in Japan. And then he moved to from research to company because probably he want to try to get more money to don't pay, but then probably he didn't get too much and returned to research to San Raffaele Institute. And after a postdoc position, he got an associate professorship at the University of Pavia, and then at the same time also at the Leuven University in Belgium, where basically still maintain a laboratory. And few months ago he got a full professorship here at Sapienza University. So I mean, we are glad that Maurilio can be also here at our university. The research activity of his team is to establish a new strategy of repairing skeletal and cardiac damaged muscle through stem cell technology, and one of the technology that Maurilio also developed is related to IPS. Basically, his presentation today is related to human IPS-based 3D models to reveal new therapeutic targets for Duchenne muscular dystrophy. So please, Maurilio.
So first of all, I want to thank Antonio for this nice presentation, all of you and the colleagues that they gave a fantastic talk, and I'm the last. It's always difficult to be the last. I will try to make you awake, don't fall asleep. And as you can see from my first slide, I'm back home. For the local people that know the soccer team of this city, you can guess which is my team that I support. So, much better now, more oxygen. So, I'm going to give an overview of what we have done in the last years and concentrate a little bit more in 2D and 3D models. That is also a kind of, let's say, tissue engineering and also organoid models. It's a kind of regenerative medicine because we are trying to generate in a dish what we can see in a human body. And the fact that the human IPS are available offer also the chance to work with the human model instead of the classical mice or other rodents. I'm talking about Duchenne muscular dystrophy because, as you probably heard about it, not much today, but it's the most diffuse disorder, genetic disorder for muscle, and actually is also the most severe. And as you can see from this slide, the fact that 90%, 95%, yes, this is the mouse, get the cardiac complication is the reason why we should look at also this important aspect, not only try to cure the muscle. And since you heard about Sharagin Taibash several times from my colleagues, I bring Sharagin here with his video that show why I still believe, and maybe few people like me, that is a perfect tissue that calls for stem cell therapy. Because as you can see from here, satellite cells that you heard about from the previous speakers, they can proliferate, they are activate, proliferate, and regenerate the fiber. So if you have a cell that can bring some information or missing a gene in this system, could work. And that's the work in, let's say, 90, 2000 from Jacques Tremblay with the satellite cells. And after that, when I moved back from Japan and together with Giulio, we were trying to use alternative strategy than satellite cells. And actually, thanks to the work of Luciana De Angelis, that I hope she is in the audience and internet, they identified this mesangioblast from dorsal aorta. And we found that a similar population exists in post-natal skeletal muscle related to pericyte, and we prove evidence that we could use these cells to regenerate muscle. And it took more than 10 years with the clinical trial of Giulio to show that actually these cells can migrate from the vessel to the tissue. And this is a great advantage compared to the satellite cells. As you know, they have a very limited capacity to migrate. And this is something that we have to look using IPS technology. And in my opinion, not to try to generate satellite cells, because otherwise we will have the same problem if we think to use for stem cell therapy. But today, I don't want to talk about this, although I think still there is interest to do this. And actually, Giulio Cossu got the second ERC advanced grant, and he wants to combine gene and cell therapy. And I'm also involved in a consortium where some clinicians want to try to test this possibility to challenge muscular dystrophy related to mutation in the mitochondria. So, I want to start with the work of a brilliant PhD student. I have a chance to work, Mattia Quattrocelli, that now is in Cincinnati as a tenure track assistant professor. And he was much more interested in what at that time was called epigenetic memory, or a bias when you adopt the IPS technology, thanks to Yamanaka, and you see here also John Gardner, that they got the Nobel Prize 10 years ago now, time flies. And Mattia started to work with the mesodermal progenitor induce starting from IPS. And what he found, if you generate IPS from a fibroblast or from the mesangioblast that have myogenic capacity, you will have a bias on myogenic differentiation. And also taking care about the progenitor, cardiac progenitor, muscle progenitor, he found a triple positive population that still can generate myotube and myofiber in vivo, but also cardiac tissue, as I show here in this movie. And what happened after Mattia was leaving, we tried to understand if the bias I was mentioning, we can capture in somehow and try to develop technology. And using a high throughput system, moving from somatic cell to generation of induced pluripotent stem cell, and the information about the signature, we could pick up some microRNA. Actually, the cocktail to force myogenic differentiation was based on 5 microRNA and 5 antagomers. And you can see here just a simple image. When we use fibroblast MIPS, we call MIPS because they are mesodermal progenitor and still able to, as I showed before, generate not only muscle cell but also cardiac cell, in particular cardiomyocytes. And you see here when we treat the fibroblast MIPS that they contribute much less. You see it here because they are GFP positive. So the green fiber are coming from transplanted exogenous MIPS. They can have the same potential of the much myogenic MAPS, as you can see here. And vice versa, if you use the opposite signature, you can prevent mesangioblast-derived MIPS, and now you see here plus anti-myogenic cocktail, you have the same engraftment of fibroblast-derived MIPS. And this is, I think it's quite important because I guess this heterogeneity that we heard before, it's even more present in IPS technology. Another point that we did in last years was the fact that during regeneration, you have an impaired situation in a chronic situation. We work in an animal model for sarcoglycanopathy, and with a single cell RNA-seq analysis, we found that a part of the satellite cells here, we have this interstitial cell population where you can find all described, let's say, interstitial stem cell, including FABs that were mentioned by Tom Rando before, but also mesangioblast here, suggesting that probably they have a similar signature, a common progenitor. And actually, the interesting point, when we use this single cell RNA-seq, this is a SMART-seq2 technology, even we were not so excited about that, we found also the quiescent satellite cells, so the PAC7 positive, myodin negative, and that was the reason that we started collaboration with a group in Paris, and they found the fantastic signature of quiescent satellite cells with the work of Leo Machado that moved to Leuven and worked together to Jordi Camps in my lab and under the supervision of Philippos and Frederic Relais. More recently, we are trying to do the same exercise with human biopsies in collaboration with Ivan Torrente from the University of Milan, and Nefele Jaratana is now the specialist of single-cell RNA-seq. In our lab, as you can see now, we improve the number of cells because we are moving from SMART-seq2 to the most probably used nowadays, 10x, that allow to have a huge amount of cells. And you can see here that with the pseudo-time, you can make a reconstruction, of the prediction of this cell in late or middle or even early dystrophic patient. And this is, I think, is quite important work that other people are doing to have not only a prediction of, let's say, the prognosis of this patient that is, again, quite variable, but also to find a novel target. That is the second part of my talk this afternoon. So, thanks to the work of Natasha Brulz, we set up the same protocol but in a chemical defined system in order to have a bank of iPS cells and progenitors in a clinical grade manner. And actually, we also prove in this way, and we adopt the same single cell RNA-seq technology to see how valproic acid, that was shown by several researchers to have an impact in myogenesis. And we confirmed that even in our MIPS, you have an improvement of myogenic differentiation. And actually, adapting the single-cell RNA-seq analysis, you can see that the treatment generates a completely shift in the cluster of treated versus untreated. And of course, the heat map tell us all the myogenic related differentially expressed genes. I just pick up some of them that we validate, like CD82, for instance, that is a marker, by the way, used for satellite cells. And in collaboration with Gunnar Beise at the University Hospital of Gustafsburg, we generate several Duchenne muscular dystrophy patients. Actually, we have also in our bank a lot of IPSA line coming from patients affected by Lingard muscular dystrophy type 2E and type 2D. However, I want to focus today the attention of this particular cell line that has a point mutation here. And as you can see from the clinical parameters, this patient developed cardiomyopathy quite early. That is the big issue nowadays of this patient. And also we managed to generate by CRISPR-Cas9 technology an isogenic control that I think is essential to prove any kind of phenotype that you can have in vitro, not only using a healthy control. And this is the work of Robin Doolan that we recently published. You can see here the absence of dystrophin when we adopt the 2D protocol to generate cardiomyocytes in vitro. That it was back when we correct with the CRISPR-Cas9. And this is quite obvious experiment, but I want to tell you that the efficiency in our hand was very poor. That's cast some doubt how this kind of CRISPR-Cas9 direct gene therapy can work. I think you really need to use lentiviral vectors or other system to really increase the chance to get the point mutation convert in a wild type. And what we have done then to prove with a collaboration with Eschenhagen in Sweden, in Germany, we did also a 3D system. Why 3D? Because one of limitation of IPS technology, not only in the skeletal cardiac muscle, but in neuro, in any kind of differentiation protocol, is the maturation, as you know. And indeed, using a Eschenhagen protocol with our MIPS protocol to generate this mesodermal progenitor, we can improve the differentiation. I have a problem with the microphone sometimes, maybe it's too close. Let's try it this way. You hear me like that, right? So, and by also our RT-PCR, we can prove that late cardiac marker you see here in a panel C are much increased when you go from 2D protocol to 3D protocol. Another strategy that we use in the lab is microfluidic. This is in collaboration with Karen Zipito, but also with Giancarlo Forte, that is in Brno University, because the extracellular environment really forces the cardiac differentiation, so the stiffness is very crucial. However, and I want to show also the negative results in our team, in particular of this French postdoc, Guillaume Gilbert, because he was struggling to generate a human cell model for ling-girdle muscle, anyway, sarcoglycanopathy maybe is better, type 2D, because unfortunately, whatever we used, 3D Eschenhagen method, forcing with microfluidic, we never detect the beta-sarcoglycan in our wild-type cells. And we published these negative results, and now Guillaume is back to France. Although after 1 year of his that was back in France, with the work of Yoke Chin-chai, we start to follow a little bit Hans Clever's strategy. Of course, it never allow us to call this kind of 3D protocol organoids, because as you know, organoids coming from adult stem cell and of epithelium origin, and when we use IPS, we are a little bit far from the original protocol of Kranens. Anyway, so we decided to call microcardiac tissue, and with this medium throughput microtissue self-assembly platform, we can able to cultivate cardiac microtissue for more than 100 days. And we recently got also the Hamamatsu photonics device that allow high throughput calcium transient analysis in 3D dimension. What I want to tell you, this is a quite recent experiment. Using this protocol now, we could see back, starting from IPS-derived mesodermal progenitor that I showed before, the presence of all sarcoglycan, suggesting that, as you can see here, by the CRISPR-Cas correction, even the calcium transient have a completely different pattern compared to dystrophic Duchenne muscular dystrophy-derived cardiomyocyte using IPS technology. And actually, Vittoria Marina with Yoke Chin-chai starting this PhD working with an interesting project to model the left ventricular non-compaction. And to do this, we need to scale up our system, and using this 3D bio printer called 3D BioBot, we can make a multi-layer mimicking what happened in a real anatomical cardiac tissue that you need to generate not only myocardium like I was showing before, but also the endocardium. And with this transgenic IPS line dystrophic activation of ATV2, you can force the expression and the differentiation towards endocardium or endothelial cell type. But in this, I think I have 3 minutes, something like that. I want just to give how you can use this 3D model to screen for, in this case, a non-drug, but also eventually we are already starting with some unknown blocker, and I show you one of the new target that we found. As you probably hear before, that not only during degeneration and aging, but also in other situations, ROS is very crucial. So the accumulation of reactive oxygen species is also present in Duchenne cardiomyocytes and also in our model. So we use the ROS scavenger and taluren, that, as you know, it's a read-through premature nonsense stop codon, and idibenone, that is a new drug that was, well, not so new, but was used in a clinical trial to improve the respiratory function in the Duchenne patient. I don't want to go in detail of all analysis because of lack of time, but I just point about the phenotype that we have. When we generate cardiomyocytes from IPS derived from this patient, we have a huge cell mortality that also reflects the increase of ROS in mitochondrial and general ROS. And when we treat with both ataluren and idebenone, that is this scavenger, ROS scavenger, but also force the production of ATP, we have an improvement of our phenotype. You see here the Western blot show a little bit presence of dystrophin due to ataluren effect. When we look at the many possible targets, we found that the NOX4, it was dramatically highly expressed, not only in our system, but if you go in literature, also in MDX mice that are resembling Duchenne muscular dystrophy in a murine model. And when we block with GAP-MERS NOX4, we can revert, as you can see here, the phenotype. You have a drop of ROS, and actually NOX4 was found also in other cancer as a mitochondrial energetic sensor. So to conclude, when we did the same experiment, we forced the treatment with idebenone, or we provide the missing ITP in vitro, we could revert the phenotype. If we combine ITP with idebenone, the system was saturated, so suggesting that idebenone was sufficient to rescue the phenotype in our model. So to conclude, I show that this kind of 3D model can not only give rise to study the mechanism, but also the possibility for screening of drug, and I show the effect of a combination of two drugs that can revert the cardiac phenotype in vitro. And we also found a new target for Duchenne muscular dystrophy cardiomyopathy, that is NOX4. I want just to conclude to thank Robin, Nefele, Joke, Vittoria, the sponsor, all of you for your attention. I'm happy to take any question.
Thank you, Maurizio. It's an interesting presentation that is open for discussion. Questions from the audience or from people that attended the symposium in streaming? Thomas.
I just was wondering if you'd be willing to share with us your thoughts on using iPSCs as cellular therapies in these diseases.
Well, as you know, IPS technology offers a kind of an evergreen pool, and differently from adult stem cell, that is the big advantage. But on the other hand, as you know, there are a lot of problems in terms of teratogenic. So that is the balance. What we have to do for the future, we should work much more for DNA stability, genome stability, and to look at now with this technology to develop also 3D structure organoids to see how far we can go with cells and maintain a structure. We try, for instance, to develop some protocol. I know that, for instance, Olivier Pourquet, that developed the technology to generate satellite cells from iPS, they are trying to do cell therapy. However, my impression there is that if those cells, they cannot acquire the ability to migrate, we will end up with the same problem of satellite cells. So I think the advantage is to look for different progenitor that still maintain the myogenic ability, but still the migration capacity, because to reach respiratory muscle, we have to go systemically, like Giulio did in his clinical trial. That is my opinion.
So Maurizio, I have a curiosity. If I were remembering in one of your papers, you published that sarcoglycan, the cardiac from sarcoglycan knockout mice develop a sort of skeletal muscle phenotype, changing, modulating some microRNA. Do you think that also these IPS technologies, when you try to get cardiomyocyte from patients, do you think that you have to deal before with this problem in order to generate a real functional cardiac muscle phenotype?
Yeah, you are referring to very old paper of Stefania Kripa.
Well,
you have good memory. 2011 Journal Cell Biology.
I don't remember the
So the microRNA that you mentioned is 69. That means it's actually one of the few microRNA that block MyoD. And the fact is that one of the members of this family was exactly in an intron of the beta-sarcoglycan that was abolished in a knockout model produced by Kevin Campbell many years ago. So it's true that we have to be very careful, and that is the advantage to use the single cell RNA-seq technology that doesn't give only the expression, the transcriptomic, but you can also have the long non-coding, but also the microRNA signature of those cells. In that sense, you can carefully control if everything is in place, and of course you can do in a huge population thanks to the 10x, because you know SMARTSEC2, there is a lot of lab work. You have much more information in terms of gene expression and microRNA, but still you don't have a larger number of cells. Now you can do, you can reach a million of cells. In that sense, I feel that you can be careful enough to avoid these kind of problems.
Okay. Barbara?
Thank you for your presentation. Since you talk about the relevance of matrix thickness in the differentiation of your cells, in your micro tissue experiment, did you use canonical Petri dishes, or you used some substrate with specific elasticity or stiffness to differentiate your cells?
So the question from the audience, Barbara, was regarding to the using of assembling cardiac tissue that I show, if I use a particular hydrogels or some material to guarantee stiffness. Well, this is a good point. We are now challenging for the 3D bioprinter, because in that case you need to use the bioink where your cell, the transgenic line, to move towards endothelial or cardiac differentiation, and there is very crucial. The one that I show you, it's without any material. It's just a printer, normal 3D printer, to have this mold to prepare this kind of micro wells where the cells are aggregates together and grow in suspension without attachment. So there was no extracellular matrix or whatever there.
Other questions or curiosity? Okay, if not, thank you again, Maurilio. So I think that we can close this long afternoon scientific session. I would like to thank again all our speakers and friends. So again, Tom, Rando, Pura Munoz, Feliciano Protasi, and Maurilio Sampaolesi. So really thank you very much, because we learn really a lot, and probably the take-home message, of course, is that probably you indicate the way, the path to rejuvenate, or at least to try to rejuvenate. So I think that we learn really a lot about this important topic related to regenerative medicine, or at least to maintain the functionality of tissues. So let me also thank the co-chair, Andrea Limonti, and Domenico Salvatore. I would like to thank again Silvia Misiti, the scientific director of IBSA Foundation, with whom we organize this interesting symposium. And on behalf of Accademia Medica di Roma, I would like to thank again all the attendees in presence in streaming. I saw that there are still a lot of people that are following this symposium, and I just would like to remember the next appointment related to the activity, the scientific activity of Medical Academy, which is on April 28th, where basically we will talk about l'etica della conoscenza, the ethics of knowledge. So I invited all of you to follow the scientific activity of Accademia Medica di Roma, and also on behalf of the President, Professor Vincenzo Barnaba, I thank you again, and I hope to see you soon. Thank you.
dell'attestato e che possono prendere le borse che ci sono.
Sì, per l'attestato potete intanto prendere le borse che sono sui tavoli, e per l'attestato potete ritirarlo proprio qui fuori, al banco qui fuori, mentre le persone che sono collegate in streaming lo possono ritirare, lo possono scaricare direttamente dalla chat.
Grazie.